The overall goal of this high-throughput yeast phenotyping is to screen for proteins that elicit a lasting phenotypic memory of past overexpression as a proxy for protein-based inheritance.
This method can help answer key questions in the yeast prion field, such as how many proteins can elicit inheritance of this type. The main advantage of this technique is that you can screen many proteins and pathways in an unbiased fashion.
Start this procedure by growing the appropriate yeast cells in a 96-well plate at 30 degrees Celsius as described in the text protocol. Visually inspect the cultures to confirm that the cultures are saturated. Cells should be visible by eye at the bottom of each well. Use a liquid handling robot to prepare 384 well plates containing 45 microliters of the appropriate medium per well. First, dispense Escal-Ura into the wells of a 384 well plate. Then dispense Escal-Ura plus the stressor of interest into the wells of a second plate. 20 millimolar of manganese chloride is used as the stressor in this experiment. Thirdly, dispense SD-Ura, which will not induce plasmid expression, plus manganese chloride into another plate. Load the 96-well plate containing the cultures onto the liquid handling robot and inoculate a 1 to 4 array of 1 microliter from each well of the 96-well plate into 4 separate wells of each 384 well plate filled with the different media. Immediately place the plates with cells on a microplate stacker at room temperature and atmospheric carbon dioxide. Set the protocol for a 72-hour continuous loop, measuring the optical density at 600 nanometers with a microplate reader. After the growth measurements, transfer 1 microliter per well of the Escal-Ura induced cultures that experienced protein.